All right, guys, so we left off with talking about the MPTP and the MPPP, right? Now, what I did is I kind of redrew this diagram here just so that we'd be able to see how we're absorbing certain things that we need to make dopamine and the certain drugs that we can use to interact with it, so that it's more open, so we're not having a lot of crossing over, all right? So first off, before we even go into the drugs that we're going to talk about to use to treat Parkinson's disease, let's see how we can actually synthesize the normal dopamine pathway. So how we actually make dopamine, because then after we understand the normal pathway, we know exactly where to pinpoint for certain drugs. All right, so first off, when you eat food, right, there's certain type of amino acid that's present within proteins, and that molecule is called tyrosine, but we're just going to denote that with T. So tyrosine is a specific amino acid. It actually has a benzene ring with an alcohol coming off of it. So it has the ability to act like a polar amino acid, but it's neutral in its activity. But what happens is it's coming across the actual GI tract and it gets absorbed across the GI tract, right? When it gets absorbed across the GI tract and gets taken into the blood, what can happen? The tyrosine can actually pass the blood-brain barrier, and what? It can come out here and it can move into this terminal. So this is the axon terminal right here. So we're just taking one dopaminergic neuron here. So we're taking one dopaminergic neuron out of the substantia nigra and zooming in on its axon terminal. So what happens? This tyrosine actually gets taken right into this axon terminal. When it gets taken in, it gets acted on by a specific enzyme. And that enzyme is called tyrosine hydroxylase. So all this enzyme does is it adds a hydroxyl group onto tyrosine. And when it adds a hydroxyl group onto tyrosine, it converts tyrosine into what's called DOPA. Okay? Now, what happens with DOPA? So DOPA gets acted on by another enzyme. And this enzyme is called a DOPA decarboxylase. So it's called DOPA decarboxylase. Okay? And what does DOPA decarboxylase do? DOPA decarboxylase converts DOPA, so it actually removes off a carbon. That's what decarboxylation is. It removes off the carbon in the form of CO2 and converts this into dopamine. And then dopamine is actually going to be taken up through specific transporters into this synaptic vesicle, which is present within the axon terminal. And then what happens is if this axon actually exerts an action potential, right? So if it exerts an action potential and sends down these action potentials down the axons, it opens up a specific voltage-gated calcium channel on the axon terminal. And calcium starts rushing in. So if calcium starts rushing in through this axon terminal, there's a reason why. So when calcium comes in, if you notice that there are two proteins right here. One of these proteins is bound to the synaptic vesicle, and the other one is here bound to the actual membrane of the axon terminal. These two proteins are called synaptoproteins, so synaptobrevin and syntaxin. And what calcium does is calcium comes in and as it acts as the cross-linking structure between these two and pulls the actual synaptic vesicle to the membrane in which the lipid bilayers fuse. When the lipid bilayers fuse, it forms this little bubble where it can exocytose the dopamine out into the synaptic cleft. And then what can happen? Well, dopamine can come over here and it can bind onto this dopaminergic receptor, which could either be D1 or D2, depending upon which GABAergic neuron we're at. So again, this is the actual GABAergic, but I think they actually specifically call it GABAergic neurons. We should say specifically, we call this a GABAergic neuron. All it means is that it's secreting or releasing GABA, if you remember that from before. So this will be one of the neurons that we talked about in the putamen. What happens? Dopamine binds onto this receptor, and depending upon what receptor it is, it'll determine a specific second messenger system, right? Now what can happen? After dopamine is actually acting on the receptor, well, every neurotransmitter can't stay in that synaptic cleft forever. So it has two mechanisms. One mechanism is it gets taken back up into the synaptic bulb. And when it gets taken back up into the synaptic bulb, some of the dopamine can actually get put right back in to the synaptic vesicle, right? So it can go right back into the synaptic vesicle. Some of it, though, can get broken down. Look at this little Charlie Brown looking enzyme. All right? So this Charlie Brown looking enzyme is called a monoamine oxidase type B. So this one is right here is called MAO B, which stands for monoamine oxidase, B. So this one breaks down specifically dopamine. There is a type A, and that breaks down epinephrine and norepinephrine. So now, some of this actual dopamine can be degraded. All right, so now we got the basic synthesis there, right? Well, there's one more enzyme. If you noticed, I drew this little purple little dude here with two little cutters, right? This purple dude right here is called a COMT. Okay, he's called a COMT enzyme. What does this COMT stand for? It stands for catechol O-methyl transferase. I'll just write that out. It stands for catechol O-methyl transferase. Okay, catechol just means it's a catecholamine. You have a specific ring. It kind of gives off this six-membered benzene ring with specific structures. We're not going to do that right now. But just know that it is specifically working on catecholamines. And what this actual COMT does is it it adds a methyl group. So if you see by the name methyl transferase, it transfers a methyl onto that specific dopamine, right? So if it transfers a methyl onto the dopamine, it converts into what's called 3-O-methyl dopamine, which is actually going to be set up for degradation. So he'll be degraded, okay? So now, if you look here, we know the normal way that dopamine is released, or then we know the way that he's synthesized, the way he's released, and then his reuptake mechanisms. So now what we got to do is we just got to try to target certain steps in there. Okay, so let's figure out one of the drugs that we can use to treat Parkinson's disease. One of these, the most common one, is called, the one that most people know about, it's called L-DOPA, which stands for levodopa. Levodopa you can take in tablet form. So when you take the levodopa, it can be brought in through the tablet form, and it can be absorbed right across the GI tract, right? So if we have here, I'm going to put L for levodopa. Levodopa can actually get absorbed across the GI tract. But here's where we got to be really careful. If you remember, we had the tyrosine. I'm going to get him out of the way. I'm going to put him over here, show his pathway coming up over there now. Because now we got to look at levodopa. So here's our levodopa. So what happens with the levodopa? He can get absorbed. But now look, there's two specific cutters here, two enzymes, I should say. This black enzyme right here stands for a dopa D-carboxylase enzyme. And this red enzyme over here is COMT enzyme. You already know these enzymes because we just talked about them. This is dopa decarboxylase, but it's a peripheral dopa decarboxylase. This one that we talked about up here in the purple, that's a central enzyme. It's only located in the central nervous system. So dopa decarboxylase is peripheral, and this is a catechol O-methyl transferase, which is only in the periphery. What happens is once levodopa gets in and it gets absorbed, there is these enzymes that can try to break it down or try to control its specific activity. So let's say levodopa gets acted on by the dopa decarboxylase. What did it do before? It converts it into dopamine. Okay, why is that bad? Because dopamine can't cross the blood-brain barrier. And when it can't cross the blood-brain barrier, one of the negative things about dopamine is whenever he starts accumulating, is it can start actually damaging it can cause a lot of side effects. One is it can actually cause a lot of cardiovascular complications. So a lot of it has a lot of cardiovascular complications. So it can increase your heart rate, it can increase the actual vasopressive actions. So it can do a lot of different things that we don't want. Okay? Another thing, it can even increase the side effects of like vomiting and a lot of GI symptoms. Okay, anyway. Another thing that we could have is the COMT enzyme, right? The COMT enzyme, if you remember, what did he do? He converts levodopa into what? 3-O-methyl dopamine, right? Or 3-O-methyl dopa, all right? So if we convert this into 3-O-methyl dopa, right now, what's the problem with this? Well, 3-O-methyl dopa actually competes with levodopa. So now let's draw levodopa here in purple, trying to get in across the blood-brain barrier. So let's say here he is. He's trying to move and he's trying to get in here, right? Because he can cross the blood-brain barrier. Dopamine can't. What's the problem with this 3-O-methyl dopa? 3-O-methyl dopa can actually compete with L-DOPA to get into the blood-brain barrier. So if you think about it, just giving L-DOPA isn't enough to treat Parkinson's disease because we have these peripheral enzymes that can convert it into dopamine, which decreases the effectiveness of the L-DOPA because now we don't have as much to get to the brain. Another thing is, there's another enzyme that converts them into 3-O-methyl-DOPA, which competes with the L-DOPA, so it tries to block the L-DOPA from getting into the central nervous system. So what do we need to do? When we give L-DOPA, we can't just give it by itself. We have to give drugs to block this or this sometimes. What drug can we give to block DDC or dopa decarboxylase? That drug is called carbidopa. So the drug that we could actually use to target that enzyme right there is called carbidopa. So what is that enzyme? I'm sorry, what is that drug called? It's called carbidopa. And carbidopa would inhibit the dopa decarboxylase enzyme. And if you inhibit the dopa decarboxylase enzyme, it won't convert it into dopamine. And you'll have more L-DOPA. Okay, that's a good thing. Another enzyme we could use is we could inhibit the COMT enzyme and prevent it from getting converted into 3-O-methyl-DOPA. What drug could we use to target that enzyme? That drug is called, you could give two drugs, but preferably they like to give entacapone. There is another one, I'll write it down here, it's called tolcapone. And the reason they don't prefer tolcapone is because tolcapone actually increases the chances of liver toxicity. So it's not a good one. But entacapone, remember, it can only work on this peripheral, it only can work on this peripheral COMT, not on the central. Tolcapone can act on the peripheral, and it can act on the central enzyme over here. Okay, so that's the difference between the two. All right, so we've targeted that step there, the COMT. And what drugs could we give again? We could give tolcapone, but not preferred, and entacapone. Okay, entacapone. So we've increased the bioavailability of our levodopa, right, with the entacapone, because we've given the entacapone, we've given the carbidopa. That's a good thing. But here's what you have to be careful of. And this, I want to mention this because it's very important. When you're giving L-DOPA with the combination of carbidopa and entacapone, if you think about it, what is it doing? It's inhibiting specific enzymes that try to convert dopa, L-DOPA, into less functional forms like 3-O-methyl-DOPA and dopamine, which either can't cross the blood-brain barrier, being dopamine, or tries to competitively block L-DOPA. But now that you've inhibited those, you have a lot of L-DOPA getting into the central nervous system. Why is that not necessarily a good thing? It'll treat the Parkinson's problem, but then guess what? It'll cause side effects. You know what those side effects are? It's going to cause opposite movement. So before, they had tremors and they had a hard time moving. Guess what they're going to have now? Now they're going to be moving a lot, having unnecessary movements. That's called chorea, or even dyskinesia. All right, so whenever you give carbidopa or entacapone, make sure that you try to lower the dosage of the L-DOPA, okay, because then it can cause a lot of side effects. All right, so now we talked about that. We get the L-DOPA in here. Where does the L-DOPA feed in? Right there into DOPA. So there's a transporter for L-DOPA. Here's this transporter for L-DOPA, and then it gets converted right into DOPA. And then DOPA can be acted on by dopa decarboxylase and get converted into dopamine. That's a beautiful thing. But again, what drug could we use here to inhibit dopa decarboxylase? The same one that we use down here, right? Carbidopa. No, we can't. Why? Because carbidopa only works on the peripheral DDC. So I'm going to write that down. Carbidopa, can it get over here and act on this one? No, because he cannot get across this blood-brain barrier, so he only works on the dopa decarboxylase in the periphery. Remember that. Carbidopa cannot work on the central dopa decarboxylase, only on the peripheral. Another drug that we can use actually can inhibit this enzyme, because what we want to try to do is the whole purpose of giving these drugs is to treat the symptoms. Unfortunately, Parkinson's disease is, you can't reverse it, you can't cure it. So because it's a progressive neurodegenerative disease, all you can try to do is treat the symptoms. And the only way we can do that is try to increase the amount of dopamine in the synapses. So that's what we're trying to do, is increase the dopamine concentration. So what else can we do? We can inhibit the dopamine from getting broken down by a monoamine oxidase inhibitor type B. What actual drug can we use there? We could use, let's actually write this one in green, try different colors. This drug could be called selagelene. Selagelene. Or you could use other ones too, like razagiline too. So you can use selagiline, and what selagiline does is it comes over here and it inhibits the monoamine oxidase B. So this is a monoamine oxidase inhibitor. So you can give selagiline, right? So you can give selagiline, and there's other drugs that you can also give besides that one too. So another drug you can use is called amantadine. Now amantadine is actually used as an antiviral drug, but you can also use it in the treatment of Parkinson's disease. Now the mechanism they're not completely sure of, but what they do know is that it helps to be able to increase the synthesis pathway, so the synthesis of dopamine. It also has been found to increase the actual release of dopamine. So what is the first thing? It increases the synthesis of dopamine, so all this actual tyrosine to dopa to dopamine, it increases that pathway. It also increases the release pathway, and it decreases the actual degradation pathway. So it decreases the reuptake. That's important. Let me write that down actually. So what are the three effects of amantadine? So let's say here's amantadine. The overall effect is increased synthesis, okay, increase release, and then decrease the reuptake. Why do we want to decrease the reuptake? Because we want to keep the dopamine out in that area. We want the dopamine to continue to exert its effects upon the GABAergic neurons. So we don't want it to get taken back in. So we want to decrease its reuptake so that we have it accumulating out there. That's the important thing. So that's what amantadine does. So again, three functions of amantadine is to increase the synthesis pathway, increase the release pathway, and decrease the reuptake of it so that we have more dopamine in those synapses. Okay, so we got that one. What other one could we use? Okay, we already said over here with the COMT, we could give the tolcapone, right? So we could also give tolcapone here. But again, one thing about tolcapone, as I said before, is it increases the liver toxicity. So they actually make you sign a waiver in the US to be able to use tolcapone because of the high liver toxicity. And they have to constantly be checking your liver enzyme levels, your AST and your ALT enzymes. Okay, so it's not necessarily the preferred type of COMT inhibitor, but it can be used. All right, what else could we use? All right, so we talked about a lot of these different steps here. What about right there? We could also specifically try to come up with some type of dopamine mimicry or dopamine agonist. What is that drug called? Or drugs. So dopamine agonists that you could use, it could be called bromocriptine. You could use pramipexole. You could use pergolide. So there's a lot of different drugs that you could use there, but all these drugs are basically acting as dopamine agonists. So what does that mean? You know how dopamine binds onto this receptor? Let's say that I denote all of these drugs with a blue dot. Guess what that blue dot can do? It can do the same thing. It can also act on those dopaminergic receptors and elicit the similar response as the dopamine. So that's a beautiful thing too, okay? So again, what are these drugs here called? They're called dopamine agonists. Dopamine agonists. Okay, so again, we've basically pretty much approached every single thing that we can for dopamine. What else can we do? Remember I told you that there was an imbalance, if you remember. An imbalance within specifically the acetylcholine and the dopamine levels, right? Whenever those dopamine and acetylcholine levels are imbalanced, it can throw off and cause what? Remember, it can cause the tremors and it can cause the rigidity, right? So we want to be able to sometimes control that activity of the acetylcholine nerve, all right? So if you see this green neuron is actually a cholinergic neuron, meaning that it releases acetylcholine. So if we have acetylcholine being released here, and acetylcholine is acting on this receptor, remember, whenever there's this decrease in dopamine, there's this balance and an increase in acetylcholine, and that's what causes the tremors and the rigidity. So why don't we give drugs to block acetylcholine? That's what we can do. We can give drugs like benztropine. So we can give drugs like benztropine, and you can even give drugs like trihexyphenidyl, okay? But basically all these drugs are doing is they're blocking the acetylcholine from being able to bind onto the receptor. So they're anticholinergic drugs. So in a nutshell, we've basically covered most of the drugs that you can use to attempt to treat Parkinson's disease. Now remember, here's the problem with Parkinson's disease. A lot of these drugs, they only work for a certain amount of time. Because as the disease progresses, the amount of dopaminergic neurons decrease. So the effectiveness of the drug will actually start decreasing over time, and so the side effects will start increasing over time also. That's why to be able to treat this, you're really only treating the symptoms the best that you possibly can. One other treatment that they can use, it's not very common, it is very rare, but it is becoming increasingly more in use, is they can try to use deep brain stimulation. So what they do is they put an electrode into the subthalamus. And whenever they put that electrode into the subthalamus, it exerts specific types of potentials that are trying to inhibit the inhibitory input going up to the central nervous system, your cortex, so that you have a little bit more stimulatory input coming down to your muscles. It has shown some effectiveness, but again, they don't use it that often. But again, it's called deep brain stimulation. Okay, so again, what drugs have we used? Let's recap one more time. We've given L-DOPA, right? And the reason why we gave L-DOPA so that we could have L-DOPA cross the blood-brain barrier, right, and get converted into DOPA. But what kind of drugs do we have to give to make sure that we increase its bioavailability? We have to give carbidopa, which only works on the peripheral dopa decarboxylase enzyme. Remember that. It doesn't work on the central. Okay? Another drug that we can give is we can use a drug to inhibit the COMT. What does he do? COMT tries to convert L-DOPA into 3-O-methyl-DOPA, which competes with the L-DOPA to get across the blood-brain barrier. So we give tolcapone or entacapone, but preferably entacapone, because it's less toxic than the tolcapone. And it inhibits this enzyme, COMT, so that more L-DOPA gets across. But remember, whenever you give these drugs, you have to decrease the L-DOPA dosage because it can cause side effects like chorea. All right. Once the L-DOPA gets in, it can get taken up through a transporter, and it can get acted on by dopa decarboxylase, convert into dopamine. What other drug can we use to be able to increase the synthesis, increase the release pathway, and decrease the reuptake so we can have more dopamine in the synapses? Amantadine, all right? Another drug that we want to be able to take care of here is we want to try to give a drug that can inhibit this monoamine oxidase B. Why we want to do that? Because whenever dopamine's brought back in, we want most of the dopamine to get recycled and get released again. We don't want it to get broken down. So we give them a drug called selagiline or razagiline, right? And what that drug does is it inhibits the monoamine oxidase B. So it's a monoamine oxidase inhibitor. And so a lot less of the dopamine gets broken down and more of it gets into this actual vesicle to get released, which is a good thing. Other drugs that we can give is we can try to be able to control the balance. Remember, there was an imbalance within the acetylcholine and the dopamine, so we can give drugs to be able to inhibit the acetylcholine response. And that drug can be called benztropine. You can also give trihexyphenidyl. Again, it's just basically blocking acetylcholine so that try to bring the balance from acetylcholine and dopamine, trying to bring it back to the normal baseline, okay? And again, you can give other drugs to be able to act as dopamine mimics or dopamine agonists. And these drugs are bromocriptine, pramipexole, pergolide, and basically they're exerting the similar effects as dopamine on these receptors and trying to elicit the same intracellular response. Okay, and you can also, if you need to, you can give tolcapone because it can work inside the central nervous system on the COMT enzyme, and the entacapone can't. And again, but you got to be careful because tolcapone has liver toxicity, so they have to actually sign a waiver and monitor the liver enzymes very, very carefully. Okay, and again, because Parkinson's disease is a very, very progressive disease, deep brain stimulation sometimes is becoming more incorporated into the treatment, where they put an electrode into the subthalamus, which exhibits these action potentials to inhibit the inhibitory impulses coming from the basal ganglia up to the cortex. So if you remember, let's come over here real quick. Remember, we had these inhibitory impulses coming up from the thalamus up here. What we can do is we can try to put an electrode here in the subthalamus, which tries to be able to inhibit these inhibitory action potentials going up to the brain, right? So it tries to be able to provide a little bit more stimulatory impulses coming to the muscles and prevent some of those tremors and rigidity and the bradykinesia or akinesia, okay?